Get started here. I want to thank you all for joining us for the U of M Conservation Sciences seminar here, and also thank Dr. Joy Bernhardt for joining us as well. We're very lucky to have her today. I'm going to go ahead and just give a brief introduction to Dr. Bernhardt, who is the Hutchinson Postdoctoral Fellow in the Department of Ecology and Evolutionary Biology at Yale University. She is an ecologist whose research aims to advance our fundamental understanding of the drivers of biodiversity change and the consequences of these changes for human well-being. Her research advances a solution to this challenge by studying the processes that unite all of life on Earth, the metabolic processes by which living systems uptake, store, and convert energy, matter, and information from their environments to grow and persist. Through this, she combines theory, experiments, and synthesis to study how living systems change as the environment changes and what these changes mean for human well-being. If at any point during the talk you have any questions, please feel free to put those in the chat box and we'll address those at the end of her talk. And then also, if you want to unmute yourself at the end of the talk and ask a question directly, always feel free to do so. Dr. Bernhardt, thank you so much for joining us. I'll go ahead and hand it off to you.
Okay, thanks. Thanks so much, Kieran, for the invitation to be here, and thanks to everyone here for joining in. I'm really happy to be here and look forward to chatting more. Can everybody see my screen all right and everything? Yeah, okay, great. So I just want to start by telling you that I'm joining you today from the Pacific coast of Canada, which is on the traditional, ancestral, and unceded territory of the Coast Salish peoples, including the Squamish and the Tsleil-Waututh nations. And I'm really fortunate to live here, and many of the scientific insights come from the amazing biodiversity that's contained within these traditional territories. So I'm going to start my talk today with where our field began. And a grand challenge in ecology over the last century has been to understand patterns in the abundance, distribution, and diversity of life on Earth. And even though we've been tackling these questions now for centuries, I would argue that we still lack a coherent and mechanistic understanding of what drives these patterns. And now we're living in an era of rapid biospheric change. And in the context of this planetary scale change, ecologists are now increasingly challenged to not only understand current patterns of abundance, distribution, and diversity of life on Earth, but also to project future ecological states as the environment changes, including what those changes mean for human well-being. So in some senses, I would argue that the core mission of ecology is changing this century from a science focused primarily on understanding patterns to one that is now focused on making projections and extending our understanding of what ecological change means for human well-being. And so I would argue that maybe the central question in ecology over the next century is to answer this question here, which is how do living systems, including the human dimensions of those systems, change in the face of environmental change? And this is the guiding question in my research program. But the problem here is that the foundations of ecology were built to understand patterns of species distribution, for example, not to project change, and certainly not to connect this understanding to human well-being. So let me illustrate this problem to you with an example. So I was trained originally as a community ecologist, so I'll illustrate you this problem with a classic model in community ecology, which is the Lacca-Volterra competition model. Hopefully you remember this model from an intro ecology class, but if you don't, the key thing to know is that this model was originally intended to predict the pattern and the outcome of interactions between two competing species. And it was originally intended really to apply to a single environment. So the question then is, how can we use a model, this sort of classic foundational model in ecology, which was built to understand patterns in the context of a changing environment? So for example, how can we use this model to predict how the outcome of competition might change as the temperature changes? Well, one thing that we might sort of start to think about is if we're trying to understand how the outcome of interactions between two competing species changes as temperature changes, we might need to know something about how those two individual populations are changing as the temperature changes. And if we want to know how each individual population is changing as the temperature changes, we might actually need to know something about how each individual within each of those populations is changing. And so what you can see here is that in order to make predictions in changing environments, we often need to understand processes operating at lower levels of biological organization. And this is actually what I think of as a general feature of developing a mechanistic understanding of change in ecology and evolution. That is, that it often requires connecting understanding of processes operating at other scales. And so this is what I do in my work, is that I aim to connect understanding of processes operating across all of these scales, from individual cells to populations to communities and ultimately up to human well-being. And I do this as a quantitative ecologist. And as a quantitative ecologist, I use a variety of tools and approaches to generate a more coherent and mechanistic understanding of change. I harness and develop mathematical theory, which I use to develop hypotheses about the mechanisms by which biological systems respond to change. And then I test these hypotheses in two main ways. The first is by using carefully designed experiments in the lab and the field. And the second way I test predictions from theory is by synthesizing large-scale data sets from a variety of sources that cover large scales, both in terms of time and space, but also in terms of taxonomic diversity. And then I harness data science tools like simulation modeling to turn all of these data into new knowledge that I use to test predictions from theory. And a common theme to all of my work is that I aim to contribute to a culture of open science. I make all of my data and code publicly available and contribute to the development of open source software tools. And I also lead training initiatives and collaborations with various communities to share these tools with a wide range of people. So today I'm going to present to you three sort of grand challenges that fall under this mission of projecting responses to global change. And I'm going to offer to you solutions to these challenges, which rely on connecting understanding across these scales. So the first challenge that I'm going to talk about is projecting the consequences of aquatic biodiversity change on human nutrition and health. The second challenge is how can we project the dynamics of populations in a warming world? And the third challenge relates to tackling this problem of generating a general understanding of living systems in fluctuating environments. Okay, so the first challenge is projecting the consequences of aquatic biodiversity change on human nutrition and health. So one of the greatest challenges facing humanity is securing a sufficient and nutritious food supply. And for many of the world's 7 billion people, food security is an ecosystem service or a benefit that's provided by aquatic ecosystems. And I'm showing you this map of the world here to draw your attention to the coastal regions, which are in this box in the orange here. These are regions of high and increasing rates of micronutrient deficiencies. They're also areas where there's a high reliance on seafood for nutrition. And these are also areas of high rates of marine biodiversity change. And so this raises the question then of how could biodiversity change affect human health and human well-being? And of course, we've known for a long time that diversity in the diet is important. This has been long recognized in the field of human health and nutrition science. And we also have models and observations that are revealing rapid biodiversity change in these areas. But quantitative and mechanistic links between biodiversity change in the ocean and human health outcomes have not yet been established. And I would argue that's primarily because these two fields have operated primarily independently, and attempts to bridge the gap just haven't been made. So that's what I did here. So, but when it comes to understanding human health benefits from seafood, it's not just a matter of understanding yields. And that's because aquatic species are valuable not only as a source of calories and protein, but also because their tissues are really rich sources of micronutrients and essential fatty acids, which are scarce in the human diet. And often when people don't have access to them, they lead to a problem that we call hidden hunger, which is when people have access to sufficient calories, but insufficient access to these essential micronutrients. And so we can measure the nutritional value of a particular fish species by measuring the concentration of these nutrients within fish tissues and comparing them to something called recommended dietary allowance, or RDA. And if you've ever looked at the side of a cereal box, You're probably familiar with RDA. RDA is a quantity of each nutrient that's required to meet health requirements of a particular demographic component of a human population. So here we answered maybe what is one of the most important questions in ecology in the 21st century, which is, does biodiversity per se, that is species richness or ecological functional diversity, distinct from the identities and abundance of any particular species, do those dimensions of biodiversity enhance human well-being? And here we can draw on a really well-established body of theory from ecology called biodiversity ecosystem functioning theory, from which we have lots of evidence, both theoretically and empirically, that increasing species richness tends to increase ecosystem functioning. And we can actually quantify the strength of the biodiversity effect here, the benefit of biodiversity in terms of ecosystem functioning, by fitting a power function to this relationship. And we can estimate the biodiversity effect or the magnitude of the biodiversity effect by estimating the value of the scaling exponent B here. So we did this here. We drew on this really well-established body of theory, and we applied it in the context of human nutritional benefits from seafood. And so here's what that looks like. So we can imagine two scenarios of levels of aquatic biodiversity in the diet, a scenario of high biodiversity in the diet and a scenario of low biodiversity in the diet. And we predict then that if species vary in their nutritional profiles and their nutrient concentrations are negatively correlated or they trade off among species, such that some species are high in some nutrients and while other species are high in one and low in another, then this could create a complementary distribution of nutrients across a range of species. And if that's the case, then biodiversity ecosystem functioning theory would allow us to predict that this should lead to enhanced nutritional benefits. So we developed two new metrics of nutritional benefit here that I'm showing you in this middle panel. The first is the number of nutrients per 100 gram edible portion. So this would count up whether you reach a calcium RDA target plus an iron RDA target plus a fatty acid RDA target, all per 100 gram portion. And we would expect that if species richness increases, then per 100 gram portion, we would expect to reach more nutritional targets. We also looked at another metric of nutritional benefit that we're calling minimum portion size. And this captures the portion size or the minimum portion size required to reach a given nutritional benefit. And again, based on biodiversity ecosystem functioning theory, we would predict that increasing species richness should lead to lower minimum portion size. And that's a benefit from the perspective of human nutrition, because if we can reach our nutritional goals more efficiently, then that's a good thing. If that's the case, then at the population level, we expect that if human populations have access to higher levels of biodiversity in the diet, this should mean that more people in the population are able to reach their nutritional requirements, and therefore more of the human population exceeds recommended dietary allowance. So this was our prediction. We set out to test these predictions by compiling a very large database of nutrient and contaminant concentrations, as well as ecological traits in over 800 seafood species. And the data looked like tissue nutrient concentration per 100 gram. We restricted our analysis to only wild species, for which we could identify which body part was included in the edible portion, and also species for which we could identify them to genus and species. And anytime you compile a very large database like we did here from a variety of different sources, you need to pay attention to quality control, and we and all of the data points here meet international guidelines for food composition, quality control, both in terms of the way that the data were collected and also in terms of the way that the data were processed. So with this large data set in hand, we then simulated seafood diets following a design called a replacement design that comes from biodiversity ecosystem functioning experiments. So what this replacement design means is that it means that species abundance in the mixtures decreases proportionally to as species richness increases. And this is an important design to take because it means that we can isolate the effect of species identity from species diversity. So with these simulated diets using a replacement design, we then estimated the biodiversity effect using this power function as shown here. Okay, so what did we find? Well, let me first draw your attention to this graph on the left, which shows minimum portion size required to reach a given RDA target. And we'll start with this yellow point and yellow line. So this yellow line here is for protein. And you can see this line is completely flat, meaning that minimum portion size is independent of species richness for protein, meaning there's no benefit at all of increasing biodiversity in the diet to reach protein requirements. But that's in contrast to the micronutrients and the fatty acids, which are shown in these other colored lines here. And you can see that each one of these has a negative slope. And I'm showing you this B parameter here, which quantifies the magnitude of that negative slope. So for all of the micronutrients and the fatty acids, we see a negative slope, meaning that increasing biodiversity in the diet reduces the minimum portion size required. This is a benefit from the perspective of human nutrition because it means that people can reach their nutritional goals more efficiently. And this effect is even stronger when we consider the 5 micronutrients and fatty acids simultaneously, which is critical in the context of human health, because of course, human health is not about reaching just one of these nutritional targets, but all of them together. And this is consistent with what's called in the biodiversity ecosystem functioning world, a multifunctional benefit of biodiversity. And so here we're showing that the multifunctional benefit, when we consider all micronutrients and fatty acids together, is significantly stronger than for any single nutrient at a time. We also found that increasing biodiversity in the diet leads to a higher number of nutritional targets met per 100 gram portion. So this is a positive relationship between increasing species richness and number of nutrient targets met in a standardized portion. So this is another benefit of biodiversity that we're showing here. And one of the most exciting findings, I think, from this work is that we discovered that the reason why we don't see a benefit of biodiversity for protein, and we do see a benefit for the micronutrients and fatty acids, has to do with the underlying trait distribution of these nutrients within fish assemblages. So I'll show you first this graph on the left, which is a fairly symmetrical distribution, and this is for protein. In contrast to that, we have calcium, which is highly skewed, meaning most species contain relatively low amounts of calcium and some contain very, very high. That means that increasing biodiversity in the diet means you're more likely to include one of these high-performing species, and that leads to a biodiversity effect. But what's really exciting here is that now that we've identified these 2 different types of distributions that exist for protein versus the micronutrients, we can then draw on a really large body of research in ecology called trait-based ecology that studies these sort of trait distributions in communities. We can then connect our understanding of what drives change in trait distributions to potential changes in the benefits of biodiversity that people get from seafood assemblages. So we also found that nutritional benefits are not only associated with species richness, but they're also related with another dimension of diversity in ecosystems, which is ecological trait diversity. So what I'm showing you here is that the probability of reaching 5 micronutrient and fatty acid RDA targets simultaneously, which is shown in this orange line, increases with ecological functional diversity. So that means that aquatic ecosystems that contain a wider range of ecological functional traits are also more likely to provide enhanced nutritional benefits. Okay, so to summarize what we found here is that biodiversity is critical to meeting micronutrient and fatty acid requirements, but not for protein. And not only that, it's essential to meeting nutritional requirements efficiently, meaning reaching targets while reducing total biomass consumption potentially. And this is really important when we consider meeting broader sustainability goals, for example, the UN sustainability goal to reach zero hunger, as well as life below water. We show that protecting, minimizing biodiversity loss in the oceans will help us achieve sustainability goals, both for human well-being as well as ecosystem health simultaneously. If folks are interested, I'd be more than happy to chat later. We also explored these patterns at local scales by looking at biodiversity effects within traditional indigenous diets in North America. And we also looked at biodiversity effects on contaminants, which I don't have time to get into here, but I would be more than happy to chat about later if folks are interested in those aspects. Okay, so I now want to move on to the second challenge. Now that I've shown you that biodiversity and biodiversity change is likely to have implications for human well-being, how do we connect our understanding of environmental change to biodiversity change? Well, one critical aspect of this is projecting population persistence in a given location as the environment changes. So a challenge here is how can we project the dynamics of populations as in the context of a warming world? And the really critical question we need to answer is how will population abundance and distribution change as the environment changes? So one way we can answer this question is to start by thinking about how metabolism might change as the environment changes. And one feature that's shared by all of life on Earth is metabolism, right? All living things must uptake resources from their environment. They must then transform these resources within their cells and then allocate the products of this transformation to the various processes underlying maintenance, growth, and reproduction. And the rate at which all of these processes occur, as ecologists, this is what we call the metabolic rate. And one of the most widely observed patterns that we see among ectotherms is that metabolic rates consistently increase with temperature across a really, really wide range of taxonomic diversity. The interesting thing here is not only is this signal apparent across a really wide range of taxonomic diversity, but also the slopes of these relationships consistently reflect the temperature dependence of cellular respiration. So what this suggests then is that constraints that are operating within cells might scale up to entire ecosystems. And this is what we call metabolic scaling, when constraints that operate at fine scales, like within cells, emerge in higher order processes. And so, but any sort of attempt to scale up from the temperature effects on subcellular metabolism to community and ecosystem scales have to assume a relationship between temperature and carrying capacity. And the reason for that is that population carrying capacity is a key parameter in many, many of our food web models. And so if we want to make projections at the community level, how food webs might change, we need to know something about how population carrying capacity changes. This has not been tested empirically in the context of metabolic scaling theory. And so in the absence of any empirical evidence, folks have either had to assume that population carrying capacity increases with temperature, or maybe it decreases with temperature, or maybe it doesn't change at all. But none of these predictions have been tested empirically. So we went about doing that. And the way that we can make predictions for how population carrying capacity should change as temperature changes is by tracing population demography back to energy budgets of individuals. And what I mean by that is that population growth is essentially an energetic process, right? Individuals, in order for a population to grow, individuals within that population must acquire more resources than they need for maintenance and then convert those excess resources into new individuals. So if we understand how individual organismal metabolism changes with temperature, this allows us then to make predictions of how population growth and population abundance might change with temperature. So I show you this model here. This is a logistic growth model, and we use it to estimate population change as a function of population growth rate, which is this little R parameter, and carrying capacity, K. So we can then draw on this prediction from metabolic scaling theory that connects our understanding of how individuals respond to temperature to making predictions of how populations respond to temperature. And the prediction here I'm showing you here is that as mass and temperature increase, metabolic demand at the individual level increases. This should lead to lower carrying capacity. And the color codes in this sentence here correspond to the terms that I'm showing you in this equation here. So the key prediction is that as temperature increases, individual organismal metabolic demands should increase. And you could imagine that if resource supply is held constant and each individual is requiring more resources, then this should lead to lower population abundance, simply because individual metabolic demand is higher. The population and resource supply is held constant, the population will be able to support fewer individuals. So this is the prediction from metabolic scaling theory, and I'll just show you what that looks like graphically here. So as temperature increases from left to right, if individual metabolic demand increases, then based on this theory, we should be able to quantitatively predict a change in population abundance as temperature increases, and this slope should be quantitatively predicted based on the slope of individual metabolic demand as temperature changes. So this prediction had not been tested empirically, and so that's what we set out to do. So we did this using a fairly simple experimental design using a species of phytoplankton that was isolated not far off the west coast of Vancouver Island in Canada. This was done in parallel with an awesome team of undergrads who all developed their own sort of independent projects in parallel with this. And I don't have time to tell you about those, but if you're interested, I would be happy to tell you about those projects as well. But the experimental design was fairly simple. We established populations across a temperature gradient, and we monitored them until they reached carrying capacity. And I'm showing you this video here of the species Tetraselmis tetraheli because I want to show you that this is a highly motile species. You can see it swimming around there. And what that means is that it allows us to meet the assumptions of a logistic growth model, which is that each individual has equal access to resources. So, but the first step to testing this theory is that we need an estimate of organismal metabolic demand. So metabolic theory would predict that we would see an increase in organismal metabolic demand as temperature goes up. And I actually measured organismal metabolic demand in phytoplankton by putting them in tiny, tiny little well plates and measuring how much oxygen they consume and how much oxygen they produce as a result of photosynthesis. And we see, consistent with many, many observations, that organismal metabolic demand increases with temperature. So now that we have a quantitative estimate of how individual metabolic demand increases with temperature, this should then allow us to make a prediction based on theory of how carrying capacity should decrease with temperature. So here's our theoretical prediction shown on the left, and here's what we found. So what you see here is carrying capacity plotted on the y-axis as a function of temperature on the x-axis. And our observations here are shown in these circles. So what you can see is that carrying capacity does decline as temperature goes up, but not nearly as much as theory predicted. So the theoretical prediction is shown in this dotted line here, and our observations are shown here. What we see is that at the warmer temperatures, populations were able to maintain higher abundance than theory predicted. So our attempts then to scale up our understanding of organismal metabolic demand as temperature changes to a population level outcome didn't work, right? So either the model is wrong or there's something going on that we didn't account for here, right? So one thing that we observed over the course of this experiment is that body size decreased with warming. And so I'm showing you here a plot of cell size as a function of temperature. And in retrospect, this is actually not a surprise at all. This is also consistent with a really widely observed pattern among ectotherms known as the temperature size rule. And in the temperature size rule, ectotherms tend to get smaller as temperature increases. So, but our original theoretical predictions did not account for the temperature size rule. So I'll show you back our original theoretical predictions, which are that as mass and temperature increase, metabolic demand should increase and carrying capacity should decrease. But what you can see here is that mass and temperature are totally independent here. This theory does not allow for any phenotypic change in mass itself as temperature changes. So it essentially completely ignores the temperature size rule. So what we did here then was to expand this theoretical prediction to allow mass to change as temperature changes in this really common form of phenotypic plasticity known as the temperature size rule. So now our new theoretical prediction are similar, but they allow us to account for this temperature size effect, which is captured in this term beta. So we now have a new theoretical prediction that incorporates estimates of temperature effects on metabolic demand, but also on individual body size. And when we look at our data in the context of this new theoretical prediction, we find actually that the temperature dependence of equilibrium abundance or carrying capacity does reflect the temperature dependence of organismal metabolic rates, in this case, the temperature dependence of photosynthesis. So now I'm showing you our new theoretical prediction that accounts for the temperature size rule in this dashed line, and you can see that this predicts the decline, the observed decline extremely well. So what this means now is this is the first evidence that changes in population abundance with warming are mediated by individual metabolic rates. This is a very, very powerful finding, I would say, because it allows us to link our understanding of how individual organisms respond physiologically to changes in temperature and extend that understanding up to population levels, and ultimately allow us to make projections of population persistence as the environment changes. Okay, but I want to point out that any form of projection of population persistence in changing environments needs to consider evolution. And the reason for that is that we have lots of evidence now that evolution can occur quite rapidly on ecological timescales. And so if we're trying to make projections then of how populations will change as temperature changes or any other form of environmental, any other sort of abiotic dimension changes, we need to consider the potential for rapid adaptation in these populations as the environment changes. So I've now shown you that metabolic traits are critical to predicting population level outcomes as the environment changes. And now I want to answer this question of how do those metabolic traits themselves evolve as the environment changes, particularly when the environment changes quite rapidly. And so what I focused on so far is the effects of temperature, but of course we know that concurrent with changes in temperature will be changes in resource supply, right? Nutrient supply regimes in the ocean will change as temperature changes. And so here we address this question of how do metabolic traits evolve or adapt in rapidly changing resource conditions? And so to address this question, we used experimental evolution. And here we're simulating rapid changes in resource supply rate. And we considered 7 different resource environments, which I'll tell you about in a minute. In particular, we focused on adaptation to low nitrogen, adaptation to low phosphorus, and adaptation to low light, and then a few others. And over this course of experimental evolution, which lasted about 285 generations, after that period of experimental evolution, we quantified evolutionary change in a key trait that we call the minimum resource requirement. This is otherwise known as R star. R star is a key trait that links individual physiology to the outcome of competition among species. So here we asked, to what extent does R star change in rapidly changing resource environments as these populations adapt to these changing conditions? We expected going into this that we would observe adaptive trade-offs. So this is widely predicted by evolutionary theory, and I'll show you what I mean by that here. So if we consider change in 2 trait dimensions, and in this case, a lower value is better, right? Because the trait that we're considering here is minimum resource requirement. From a population's perspective, the lower their minimum resource requirement, the better it is, the more likely they are to win in a competition, for example. So what would trade-offs look like in this context? Well, if were we to observe trade-offs, what it would look like is a change in one trait dimension, so getting better in one dimension, would come at a cost. Sorry, getting better in one dimension would come at a cost of getting worse in another dimension. So what that would look like is a trade-off where we would see points improving in one of the 2 dimensions at a time, not simultaneously. And that would look like a negative relationship here, were we to plot it on 2 axes. So we predicted a negative relationship, we predicted trade-offs. What we found is completely the opposite. We found no evidence for adaptive trade-offs. Instead, what we observed was a positive relationship where multiple competitive traits improved simultaneously. So what I'm showing you here is a change in light requirement plotted against a change in nitrogen requirement. And what you can see here is that many of the populations after this period of experimental evolution improved in both of these dimensions, lowered nitrogen requirement and lowered light requirement. This is completely unexpected from theory, and this is one of the assumptions that many of our theoretical predictions make. And we think that we might be seeing this here because when it comes to these essential resource traits, these are all connected via a metabolic network. So what I mean by that is, for example, if you are a population forced to grow under low light conditions, you might then adapt by producing more efficient chloroplasts, and because chloroplasts are the light harvesting mechanism. And if you can produce chloroplasts more efficiently, chloroplasts are primarily made up of nitrogen. this could actually also simultaneously reduce your need for nitrogen. So what I think is going on here is that in rapidly changing resource environments, there's actually quite a lot of room to make improvements simultaneously. And this is because in very quickly changing environments, there's potentially quite a ways to go before species reach some sort of optimality frontier. And this is a concept borrowed from economics known as the Pareto efficiency frontier, Pareto optimality frontier. And this is a point where improvements in any one dimension will come at a cost of improvements in another dimension. And so what I think the important finding here is that when we consider adaptation in very quickly changing environments more generally, we need to consider that some of those adaptive trade-offs that we predict we might not see, simply because there's still quite a ways to go before these populations reach some sort of new fitness optimum, at which point they would be facing these hard constraints and hard trade-offs. So the key point here is that this is empirical evidence that we might need to rethink some of our fundamental assumptions, especially when it comes to rapidly changing environments. Okay, so I now want to just move to the final section of the talk, which is projecting species distributions in variable environments. And one really common way of making projections of species distributions in variable environments is to scale up from thermal performance curves or estimates of measures of organismal performance generated in lab conditions up to potential species ranges in nature. And some work done about 10 years ago by Jen Sunday came to this really important finding, which is that species distributions often cannot be predicted from estimates of the fundamental niche measured under constant lab conditions. And so what this looks like then is that if we attempt to predict species ranges based on these fundamental thermal niches measured under constant lab conditions, we sometimes see cases of underfilling and sometimes cases of overfilling, these sort of mismatches between projections from what we estimate in the lab compared to what we observe in the field. So this was a really important finding and launched all kinds of interesting explorations into what could be causing these discrepancies. I mean, what explains why the thermal niche cannot predict species distributions in the field? So for example, questions got raised, could it be species interactions? Could it be other niche dimensions? Maybe it's water availability, other things other than temperature that are limiting species distributions. But I would argue here that there's maybe a much simpler explanation for this, which is that population predictions for population persistence and species ranges must explicitly consider environmental variation. And the reason for that is that organismal performance curves are almost always non-linear. And we need to explicitly consider the non-linear nature of these curves with environmental variation if we want to make accurate predictions. And this is because of something called Jensen's inequality, which is a tenet of nonlinear averaging that tells us when we're dealing with any sort of nonlinear function, in this case, as temperature varies over time, we can't project performance in variable conditions based simply on the performance in constant conditions, because this is a nonlinear function. And specifically, we expect variation to increase performance when this curve is accelerating here, which is in this blue chunk, or it should decrease performance relative to constant conditions when this curve is decelerating. So we set out then to test some of these predictions empirically by asking simply, can the temperature dependence of performance be predicted under fluctuating conditions if we know the shape of the curve under constant conditions and we account for these effects of nonlinear averaging. So we asked then, does thermal variability alter the realized thermal niche? So we set up experiments where we grew populations under constant conditions and then also under thermally fluctuating conditions, using a similar experimental setup that I showed you before here with the species of green algae again. So when we did that, we found that temperature variability systematically and predictably alters the realized thermal niche. This is not surprising based on math, that if you take a thermal performance curve like this one shown in green, and you use nonlinear averaging to project population performance in variable conditions, we would predict what we see here in the dashed line. you can see that this whole curve is actually shifted down and to the left. So the entire thermal niche is shifted to lower conditions. This is what we observe empirically. We were able to quantitatively predict this change in the thermal niche of the population in fluctuating conditions simply by accounting for the nonlinear nature of this curve. So what we're showing here is that as soon as you introduce any form of variation in the environment over time, this will systematically and predictably alter the shape of the thermal niche. And we then expanded these predictions out to the global scale by drawing on thermal performance curves from about 100 different species of phytoplankton. And we showed that when we account for natural patterns of environmental variability over time by using sea surface temperature records from the isolation locations of all of these species, we see that T-opt systematically is predicted to shift to lower temperatures than we would predict from constant lab conditions. There are a few cases where T-opt, which is the optimal temperature for performance, shifts a little bit to the right, and that's because we had a few species in our data set that showed these positively skewed thermal performance curves. So what does that then mean if we go back out and scale up to our attempts to project species ranges based on thermal performance curves? Well, what we're showing here is that accounting for natural patterns of thermal variability in nature could potentially explain discrepancies between the fundamental thermal niche measured in the lab and species distributions that we're observing in nature. So what we're showing here is a potentially much simpler and more parsimonious explanation for why we see those patterns of overfilling and underfilling that don't require any sort of other alternative species interactions or other niche axes. This is simply just a case of if we account for patterns of thermal variability, we can potentially more accurately predict species ranges in naturally fluctuating conditions. So I want to end by telling you about some of the newer directions that I'm taking in my work right now. And these have to do with ways in which living systems respond to thermal fluctuations or any form of environmental fluctuation. What I've shown you so far and what is characteristic of much of our understanding of ecology and the way that living systems respond to environmental change relies on feedbacks. And so feedbacks, I'm showing you an example of feedback here where we have some sort of environmental change depicted in this gray line and a biological response shown in the black line. Feedbacks are really common in ecology, and the sort of key component of a feedback is that some sort of change in a system, some sort of deviation from some sort of set point that's detected triggers some sort of change within the system to return back to a set point. But inherent in feedbacks is a delay because the system needs to detect the change internally and then respond. What we have proposed here is that there's potentially a completely alternative way of dealing with environmental change, which is instead of waiting for the change to happen, to actually predict or anticipate the change in advance and prepare in advance. So what that could look like is what I'm showing you here on the right. If we have some sort of environmental change that's correlated with some sort of cue that happens before the change, as long as the organism or any component of a living system can detect a cue that occurs in advance of the change, and it can internalize a correlation between a cue and a given future environmental change, then it can actually respond in the present to some expected future change. This is what we're calling a feed forward. Feed forwards are commonly studied in systems biology, and here we're extending this understanding of feed forwards to the population and community level. So I'll just give you a little bit more of an example of what makes a feedback different from a feed forward. As I mentioned earlier, feedbacks are necessarily reactive. So a nice example of that is the core body temperature of an endotherm. If there's some sort of set point, and that set point if an internal body temperature deviates from that set point in some way, then the organism will kick in some sort of physiological response to return to that internal set point. But there's some sort of delay there. In contrast, feed forward is necessarily proactive. So an example of that is this case of diel vertical migration in zooplankton. These zooplankton are capitalizing on a cue that comes from the environment, which is this, which is a change in sunlight at the surface that happens before predation pressure at the surface might increase as the sun rises and visual predators show up. They capitalize on this cue to swim down, to move to depths, to get out of the way, to therefore avoid high predation risk at the surface. And this allows them to avoid predation by capitalizing on some cue that they're using to predict the future. This is beneficial, right? Because it allows them to avoid predators, presumably increase their fitness. The way that these feed forwards arise is because organisms can exploit repeated associations between multiple different environmental variables with a time lag to anticipate change. And so I'm not sure how I'm doing on time here, but I think I'm okay. Maybe folks can just flag me if we're getting too close, because I'm not sure when we started, but I'm looking like we're doing okay.
Yeah, you're doing fine, definitely. Okay. I think we have about 13 minutes left. So.
Great. Okay, so so let me explain to you then what I mean by how feed forwards arise on this slide here. So if you can envision one environmental variable shown in this green sequence of green events shown here, and the time lag between successive green events is depicted by this tau term here. You can see that there's a fairly wide range of time lags between successive events. So there's that's shown here in this distribution. So from the perspective of an organism, this green event, which could be a change in oxygen, is really, really highly unpredictable. There's just a very, very wide range of lags. Similarly, if you look at this gray event here, this might be a change in temperature. This also has a similarly wide range in lags between each successive event. So from the perspective of an organism like an E. coli here, predicting a change in oxygen seems fairly difficult. There's just a very, very, it's kind of a random sequence of events. However, if we look at the green bar in context of the gray bar, there's a very, very tight lag between green and gray, which means that if the organism can detect a change in temperature, which could be that gray, and internalize the correlation between temperature and a change in oxygen, all it has to do is be able to detect a change in temperature. It doesn't have to predict a future change in oxygen, it just needs to detect a change in temperature in order to prepare for a change in oxygen. So in the case of E. coli, what we know is that E. coli can exploit a correlation between a change in temperature as they go from living outside on some surface to entering the human gut. When they do that, that increase in temperature is associated with an impending change in oxygen as they enter the human gut, and they can actually switch their metabolism from aerobic to anaerobic pathways to prepare in advance. So the key point here is that if organisms can internalize correlations between multiple environmental conditions, that allows them to prepare in advance, and that is a feed forward that confers a fitness benefit. And it turns out that these sort of cue-based feed forward processes are everywhere when we think about them in this way. So phenology that's cued by some sort of critical day length, circadian rhythms, diel vertical migrations, these are all feed forward processes that rely on some external cue to allow organisms to prepare in advance for some future environmental condition. And what I think is really exciting about thinking about these feed forward processes is that global environmental change is likely to change what we're thinking of as cuescapes that surround species. And as these cues change and the reliability of these cues change, this can mean really different things for co-occurring organisms that rely on different types of cues. So this is something I'm looking at now, which is how could changing cue landscapes change community responses to environmental change. Okay, so I'll end here by reminding you that I started off with this big scientific challenge, which is how can we project future ecological states in the context of environmental change. And I suggested to you a solution to this challenge, which is studying mechanisms that cross scales, connecting understanding across boundaries, particularly using established tools in new ways. And I would argue that when we do these things, this is going to advance our understanding of living systems and ultimately improve our ability to secure a sustainable future for humanity. And with that, I would like to thank my sources of funding and my collaborators, and I would be more than happy to take questions. And if you'd like to reach me, you can contact me here.
Fantastic. Thank you, Dr. Bernhardt. That was a great talk. We really appreciate you coming today. I think what I'm going to do is just open it up. So for anyone who wants to actually unmute and ask a question directly, I'll give a few seconds for someone to do that. And then we can go off into the chat and I can read out some questions to you as well. So if anybody has any direct questions, please unmute yourself and ask directly. Well, that's probably enough awkward silence. So unless somebody wants to hop in, I think I see someone unmuted.
Yeah, I guess I can ask a question. Thanks for your talk, Dr. Bernhardt. I was kind of curious about the degree of of individual variation that you saw in responses to temperature and metabolic rate. Of course, like since carrying capacity is a population variable, it probably all sort of evens out, and so you can get the theoretical predictions work out. But I'm kind of curious, like what do you think is behind all of that? The kind of scatter that you saw in that graph.
Yeah, that's a great question. And there is a lot of individual variation. I actually think that's like one of the key next sort of things that we need to be focusing on is how do we incorporate all of that individual variation that exists? How do we not ignore it? And how do we just rely on the means? That can the individual variation can be very problematic whenever those individuals are related to some population outcome by some sort of nonlinear function. Much in the same way that that I was showing you in the case of the nonlinear averaging problems when temperature varies, but of course when individual body size, for example, changes, and body size is related nonlinearly to some other process, then understanding that variation is really important. I totally agree with you. What explains that variation could be a whole bunch of different things, and that's actually something that I don't really have a good answer to. I'm working on it right now, actually, in the context of variation in thermal tolerance, and I'm explicitly looking at sort of relationships between genetic variation and variation in thermal tolerance, as well as sort of like physiological trade-offs and constraints that could shape that variation. So like a short answer, a long answer is that it really matters. A short answer is that there's probably a whole bunch of different underlying physiological processes that are contributing to that variation. And I guess I should say that that variation should be expected, and we absolutely cannot ignore it, and we need to sort of tackle it head on. And I appreciate that question in that context.
Yeah. Thanks.
That's great. Anyone else have a direct question? I see.
I can do a direct question instead of my chats. So
cool.
I'm Jerry Cohen from the Department of Horticultural Science. And so Dr. Bernhardt, I come from a different order scale from you because I worked on molecules and metabolomes. And so a couple of questions I asked. The first one was you showed protein in the early slides as being not changing, but I've worked a long time on protein quality, and especially I've been very interested in tryptophan because it impacts over 1 million children a year in terms of mental retardation, and also it affects niacin availability. And so that was not in your, the idea that it would be flat is basically, I think, not digging deep enough into protein quality in terms of an issue for human and other population dynamics, because there are components of proteins that are not uniform across all protein qualities.
Yeah.
Then the second one, which I asked the question, but then you started getting into it. So in plant biology, when we look at abiotic stress, you can't just look, for example, at a plant's ability to tolerate a certain temperature because there's acclimation. And like in the summer, you typically will see days getting hotter and hotter and hotter. They don't just get hot all of a sudden, even in Minnesota. Well, we don't really get hot in Minnesota. We just get less frozen. But acclimation, and you say the clue, which is another term for acclimation, but I think it's really an important part of how organisms respond to the environment is their ability to acclimate and how long and persistent that acclimation is, as well as how well they acclimate. So that's why your diagram of using a square wave is not a really good model of how plants respond to changing thermal stress, because it ignores acclimation as a component to that process.
Yeah, I completely agree with you, and I completely agree. Acclimation is really important when we're trying to understand responses to changing conditions, and it's something that I'm also actually working on in a couple of different ways. So, what I didn't show you was for several of the experiments that I've done, I actually tested out different acclimation treatments. So, acclimating at different temperatures for different amounts of time, and looking at how that changes responses to changing conditions. So I absolutely agree. Acclimation is really important. In phytoplankton, actually, in single-celled organisms, acclimation has very, very strange meaning, because these are single-celled organisms. And so are we talking about transgenerational acclimation? I mean, they divide on the scale of hours. So all that to say that acclimation is a really tricky nut to crack when it comes to microbes. And it's something that I am actively working on. So if you have any ideas, it would be great to chat more about that.
No, I would just say it's harder in microbes than in multicellular organisms, mainly because you have other considerations. For example, if you're looking at microbe, the vast majority of microbes, especially in an aquatic environment or a sea environment, are constrained by quorum sensing, and quorum sensing itself has a thermal character. It's because anything that diffuses has a thermal coefficient. So that you can look at Planomonas is not a good model for sea microbes because it lacks quorum sensing. That's why you can actually grow it in the lab. But that's a small amount of ocean biomass. So it becomes much more complicated. I don't have a solution for you. It's just that when you first presented it with the square wave, since we work on that acclimation aspect in terms of metabolomics and metabolic changes, we see that acclimation changes a lot of metabolic activity in order to accommodate that subsequent change in to tolerate a higher stress level.
Yeah. Absolutely.
So so I have no idea how you work on that on microbes, except to wish you all the best for a very complicated system that needs.
Yeah, thanks. We'll check back in 10 years.
Well, it needs to be solved because it accounts for such a large amount of the world's biomass.
Yeah, absolutely.
People aren't eating that, but a lot of things are eating them, and so it does make a big difference.
Yeah.
Yeah, that's great. It sounds like you guys could have some long, interesting conversations definitely in the future. So I guess, we are right at 1 o'clock here. So I did want to, there were a couple more questions in the chat here that we may not have time to get into, but I would, I guess, suggest that if anybody would like to reach out to Dr. Bernhardt, please feel free to do so over email. I think you provided that in the last slide there. And, if anybody does want to stick around for just a little bit longer and ask a question directly, please feel free. Maybe we'll do one more, offer that up. But otherwise, just want to thank everybody for joining us today. Really a pleasure to have you, Dr. Bernhardt. And we look forward to reading your papers more in depth, definitely. So.
Yeah, I'm happy to stay on the line if folks want to start a couple more questions.
Okay, great. And I think Meg, a fellow lab mate, she has a lot of interest in the contaminants. So if she's still around, maybe she can reach out and ask you directly. I still see her on here, but she was wondering if you could provide the nutshell version of the contaminants.
Yeah, for sure. So the nutshell version of the contaminants is that increasing biodiversity does lead to more contaminant exposure. And the reason for that is many of the same mechanisms that underlie the positive effects of biodiversity on nutrition also operate for contaminants, because they are distributed similarly among species in seafood assemblages. However, one good piece of news is that the effects of biodiversity are much weaker for contaminants. So the benefits of biodiversity are stronger than the negative effects in terms of contaminant exposure are. The other thing is that we didn't consider like particular combinations of species. We just looked at these things at a high level in terms of random mixtures of species, because we were really interested in the effect of biodiversity per se, not like cherry picking certain combinations. And it is absolutely possible to find certain combinations of species that have really high nutritional benefit, but also very low contaminant risk. We just didn't consider those here just because it was a little bit beyond the scope, but that is absolutely the case. Yeah, so that's sort of the short answer. I don't know if you have any other questions that you wanted to dig into on that.
No, just curious. Thank you so much.
Yeah.
That's great. So I guess, we'll let everyone go about their day and enjoy the nice weather out there. And thank you again, Dr. Bernhardt. We really do appreciate you joining us today, and look forward to seeing more of your results in the future. So Thanks again.
Thanks for the invitation.